So thank you for the kind introduction and the invitation to speak here today. I do have a conflict of interest. I have a contract with Philips. So the background of this talk, or the outline, is first talk about the background of polymers, why we want to use polymers to form contrast agents, to give some examples of polymer-based contrast agents, and also clinical examples. So polymers are essentially very large molecules. They're composed of multiple repeating units. They can be homopolymers, where the repeating unit is the same for the entire structure. And this can result in many different polymer architectures. And I've just really scratched the surface of what architectures there are available. So this is very simplified in terms of linear, branched, and cross-linked. And of course, you can have copolymers. We can have more than one repeating unit, such as PLGA, as was mentioned in the previous talk. And they can also have different configurations in terms of being random repeats of the subunits, or gradients, where there's more repeats at one end than the other, or block copolymers, which is more common, where you have all of one subunit at one end and all the other subunits at the other end. So furthermore, if you have these block copolymers, they can be in different proportions, of course, and they can have other configurations where you have, say, ABA type, where you have one polymer, another polymer, then the same polymer. If you ever use tween, or you've heard of tween, tween polymers are based on this type of chemical composition. And you can have polymers that have 3 different types, ABC, or even 4 types, and so on and so forth. And something to bear in mind if you purchasing polymers or preparing polymers, the molecular weight will vary. There'll be a distribution. So it will say 1 million or 300,000 molecular weight, but that'll just be one of the parameters of the distribution. Then if you have different blocks, then each block will have its own distribution also. So it gets kind of a little bit complicated. Okay. If you're interested in using polymers, there's lots of great commercial sources that own stock in these companies. I'm just telling you, I think it's kind of handy. Many companies other than this, and many of the polymers that have been talked about in my presentation or in previous presentations, you can buy from companies such as this. So it's very easy to integrate these polymers into your work. And as has been alluded to by some of the other presenters, there are many, many polymers. It's been a huge influence on the practice of medicine over the past, say, 30, 40 years. This is some of the agents that are used as polymer therapeutics. So you consider there are also polymeric sutures, there are polymeric implants for various applications. So many polymers are in use, and so for example, we have here polyamino acid polymers. There is a polystyrene-based polymer you see here, many polyethylene glycol-based polymers, and so forth. So this is one example of a FDA-approved product. This is Gliadel that is developed from the Langer lab and the Lyon lab. After you've had brain surgery, there's typically some tumorous cells that are remaining there in the cavity. And so the concept is you implant these polymer chips, then release a cytotoxic drug, such as carmustine, or it is carmustine in this particular example, that then hopefully kills remaining tumor cells. And this polymer is very specifically developed for this application. This degradation time is about 3 weeks. So this subunit degrades extremely quickly. This subunit polymer composed only of that degrades very slowly. So they've really tuned the ratio between the 2 polymer types to have optimal properties for the application. Okay, and a couple more examples. This is Doxil, which is a nanoparticle that's approved for clinical use, and although it has a phospholipid membrane here, polyethylene glycol is used for the coating, which is a polymer. And at least for the purposes of this talk, I was asked to consider proteins such as albumin as being polymers, which you can think of proteins as being polymers in a sense. And Abraxane has been more recently approved as another thought of as being a drug-loaded nanoparticle for cancer treatment. Okay, so why should we use polymers to form contrast agents? Well, my first reason really is that given all this use of polymers in medicine, it has to be kind of foolish not at least to try to think about it sometimes. There are many contrast-generating materials, different types of nanocrystals that really need to have coatings or frameworks to act as delivery vehicles for their use in vivo. Furthermore, if you think about natural polymers like proteins or polysaccharides, they are, of course, biocompatible, biodegradable, and they can be interesting just fundamentally to track where they're going in person or in an animal. And polymers can be formed into nanoparticles or can just simply have many of the advantageous properties of nanoparticles, such as long circulation times. Furthermore, you can exploit polymers to enhance the contrast-generating properties of contrast media. Okay, so let's just go through these points. So here are some nanocrystals. There are many types of gold nanocrystals, spheres, cubes, stars, plates. Almost you name it, people have made it. And they produce contrast for a real range of imaging techniques, computed tomography, fluorescence, photoacoustics, and they can also have therapeutic properties. So being able to use them is very useful. But say oftentimes they come with a coating, or they're initially synthesized with a coating that is not very biocompatible. Also iron oxides for magnetic resonance imaging, quantum dots as fluorophores, as was just mentioned, and also a range of other nanocrystals, bismuth, manganese, are used. So what's the issue here? Well, say one of the classical methods of synthesizing gold nanocrystals is the Turkovich method. That results in citrate-capped gold nanocrystals. And if you introduce even just phosphate-buffered saline to that kind of solution, the particles become unstable, aggregate, and turn basically into solid gold. And that is nice in certain circumstances, but if you're looking to use them in vivo, you want them to stay dispersed. And so if you apply a large capping ligand, such as a thiol PEG, so a polyethylene glycol with a thiol group at the end here, that thiol group will adhere to the gold surface, and in PBS, in blood, biological media, these particles remain dispersed. And I mean, there's really many ways to go about this. If you start off with your metal ions, you can go through a route such as the Turkovich method to end up with citrate-coated gold particles, and then do ligand substitutions. That's what I just described. You can also directly synthesize nanocrystals from, with the polymer in question to create coated particles. More recently, in the past, I guess, decade or two, nanocrystal synthesis has switched to hydrophilic methods. These often produce very controlled nanocrystal properties in terms of size. So it's often the best method to synthesize these cores. However, this is now a crystal that's soluble in toluene or hexane or something like this, and you really have to do something to make them applicable in vivo. So you can go through routes of ligand substitution, coating with different kinds of amphiphiles, or embedding in a matrix such as PLGA. So here's just a couple of examples where gold rods were synthesized, and they are coated after being synthesized with a lipid known as CTAB, which is actually very toxic. So if you substitute the coating with thiol PEG, you end up with a nice stable gold nanorod. In this study, they used the rods for, they performed CT imaging, which localized the particles, and then they performed photothermal ablation on tumors. This is a study from a few years ago where I was lead author. We used a polymer, which you can see here, which is amphiphilic. So each repeating subunit is amphiphilic that has a charged head group and a hydrophobic tail. This can be used to encapsulate hydrophobically capped iron oxides, which we then used in vivo. And you can see the contrast in liver 24 hours post-injection. There's many different types of natural polymers. We'll go into some more detail later. Proteins, polysaccharides, other things. So these are renewable, they're biocompatible, biodegradable, so there's many advantages to using these types of agents. And And if you are interested in knowing what these particles are doing, it can be interesting to track them. So here's just a couple of examples of ferritin and dextran, which is a polysaccharide. Furthermore, you can form nanoparticles in many different routes from polymers, and you can have many of the examples that some of the previous speakers talked about in terms of long circulation half-life, accumulation in tumors by the EPR effect. It's a great platform typically to integrate lots of different functionalities and so forth. And actually, if you dig back into some of the history of the literature, some of the work where the EPR effect, things like this were established back in the early 90s was using polymers. And here you have an example where we have different polymers, different molecular weights. So here is really low molecular weight polymer going up to very high. And this low molecular weight polymer is so small that it can be renally excreted. So it has a very short clearance time. Whereas these larger polymers are not renally excreted, so they have a long circulation time, and they also accumulate in the tumor. And the last thing is about enhancement of contrast. So there's several factors that affect the contrast produced, say, by gadolinium chelate. As you can see here, the factor I'm thinking about right now is the rotational correlation time. And this is data that show as the correlation time lengthens, so as the rotation slows, that affects the relaxivity, so the MR contrast produced by gadolinium chelate. And here's some real data. These are gadolinium chelates. They have a hydrophobic group on the side here, and albumin has certain pockets that hydrophobic substances can bind to. And so if these chelates bind to albumin, then the rotational correlation time decreases and the relaxivity increases, as you can see here. Okay, so let's go through some examples of some different polymer types and how they can be used to form contrast agents. So you have hydrophilic polymers, such as polyethylene imine, amphiphilic polymers, where one portion, another portion is hydrophilic, and hydrophobic polymers, such as PLGA. Dendrimers, and Pan-Am is the most frequently used dendrimer, and then natural polymers, as I say, such as ferritin. So this is a recent example from a group of Sava Bourgeois at Hopkins, and I think this is probably the most complicated molecule I've ever seen, but it actually contains 3 different types of hydrophilic polymer. So this is polylysine, this is polyethylene imine, and this is polyethylene glycol. And it has, in fact, 2 different types of targeting ligand. It has 2 fluorophores, rhodamine and Cy5. There is indium 111 attached that gives SPECT contrast, and also is complex with siRNA against a therapeutic target. And so it shows in this paper, here is some characterization via electron microscopy, where you see the particles range from around 30 to 70 nanometers. What you see here is SPECT imaging, where a mouse bears 2 different types of tumor, and one tumor bears the targeting ligand, the other tumor does not, and so you can see there's greater accumulation of the agent in the tumor that has the targeting ligand here. This is another experiment where you're looking at tumors taken from this type of mouse. It's different types of tumor. And in this instance, they injected an excess of the free targeting ligand. And you can see that the uptake of the agent is suppressed when they do that, indicating the targeting mechanism. And last, in these 2 figures here, so this is measuring the target which they directed the siRNA against, and you can see that this is reduced after injection of these particles, indicating the therapeutic effect that they were looking for. Now this is kind of an old example, but I think it's a very nice example of a block copolymer that's amphiphilic. So this section here has triiodobenzoic acid derivatives attached, and it becomes hydrophobic. And this section here is hydrophilic. It's a PEG chain again. So when these molecules are suspended in water, they form micelles, as you can see here, with a iodinated core. The iodinated core can produce contrast in CT imaging. These are CT images of mice, of rats. This is pre-injection and post-injection. And you can see the contrast here in the heart, in the aorta, in the liver, then also in the spleen. Now let's talk about hydrophobic polymers. So one of the, most famous, most widely used is PLGA, polylactic co-glycolic acid. It has some pretty advantageous properties in terms of biodegradability and controlled release. And so this has been explored as a polymer to temporarily suspend nanocrystals before the polymer breaks down, degrades, and the nanocrystals eventually release for excretion. So this is an approach that's been taken by the Shapiro group, where there is a PLGA matrix. It's been emulsified using the method that the previous speaker mentioned, in this case using polyvinyl alcohol to form the coating, and they embed different types of nanocrystals such as iron, manganese, oxide, or bismuth, as well as some dyes here, and some publications and also a poster on this work at this meeting. And so here's some of the data where they show that using SEM, you can control the size of these particles, ranging from as little as 100 nanometers to over 2 microns. And if you look here in the electron microscopy, you can see the iron oxides that are embedded in the polymer cores. So they use these particles for cell labeling. And what you see here inside the cell, these greenish or yellowish dots are the particles that have been taken up by these cells. This is a Faraday control. And here you see quantification of the uptake of these particles. So you can see, especially with these micron-sized particles, it's very high uptake. And then compared to Faraday or control, you see a very significant contrast of dispersions of these cells. In particular also because there is an increase in the relaxivity of the iron when aggregated into this polymer structure. So another hydrophobic polymer that's frequently used is polylactic acid. This polymer and other polymers such as PLGA or even albumin can be used to form microbubbles for ultrasound contrast. There are agents that are clinically approved on this type of technology. So you have an air bubble inside and a PLA shell. Also again, in this case, polyvinyl alcohol coating. This is some work from my own group where we then took these structures and put nanocrystals into the shell, or drugs or fluorophores. And you can see nanocrystals in the shell here. And we demonstrated that this has some effect or some ameliorating effect on the contrast that's produced, the acoustic contrast. Nevertheless, acoustic contrast is still being produced. And also in case of iron oxide, that produces contrast in MRI, fluorescence contrast for in case of quantum dots, and CT contrast in the case of the gold particles. And you can learn more about it if you attend this poster. Now, there's another polymer type, dendrimers, and they're a little bit similar to other polymers, but they have some unique features. So they're kind of very repeatedly branched in a fixed pattern. Number of repeats is referred to as a generation. So G0, G1, G2, G3, and so on and so forth is demonstrated here. And what's unique about them is the chemistry can be done that controls their molecular weight. So you can have a precise molecular weight with a polymer such as this. They're extremely defined compared to other polymers or compared to other nanoparticles. And most frequently used dendrimer is this PAMAM. It's regarded as being fairly biocompatible and also expresses amine groups of the surface, which can then be used to react to attach other substances. So this is an example from Tarek Fahmi's lab at Yale, where I think this is a G4 PAMAM dendrimer. And they attached polyethylene glycol to try to extend the circulation half-life. They attached some triiodo-benzoic acid groups for CT contrast, and also technetium-99 for SPECT contrast. And you can see here the contrast in the heart of the animal post-injection in CT. And this is SPECT CT, and actually you can see the contrast in the heart in this image also. But you can see the SPECT contrast localized here. Another study, in this case, they used a small dendrimer, and this dendrimer, this G3 dendrimer, is so small that it can be excreted via the kidneys. What they were looking to do was to link some of these subunits together to create a larger structure with disulfide links. And so these disulfide links can eventually be cleaved to break this up into smaller particles, which can then be excreted via the urine. And as this was a study where they're looking to incorporate gadolinium as an MR contrast agent, they were concerned about retention of gadolinium has been linked to nephrogenic systemic fibrosis and toxicity. So the idea is if this breaks up and is excreted, you avoid that type of toxicity. So what they show here is these aggregates alone are stable, and then when you add a dithiotol reducing agent that breaks these disulfide linkages, break up these particles, and also as pH is reduced, that also breaks these linkages. And here are some data where this is MR images before and then after injection. And so you can see contrast in the blood vessels and also the kidneys of these mice injected with these aggregates. And in the case of similar particles that are just sole dendrimers that haven't been aggregated, you don't really see any contrast. And when you look at the pharmacokinetics, it's clear that this G3 dendrimer has been excreted very quickly, whereas there's a longer circulation time for this aggregated dendrimer. And what the biodistribution here tells you is that it's pretty well excreted, because, say, a G4 dendrimer would probably have a liver uptake of 30-40% at this time point. So, although there's still some retained in the animal, it's pretty good excretion. Now we come to natural polymers. So we're considering here proteins as being natural polymers in this case, such as ferritin, albumin, viruses, or protamine. We covered some of this area in a review a few years ago. We have various polysaccharide-based polymers. So dextran is probably the most famous, but also heparin, alganates used a lot, also chitosan. So just start off talking about ferritin. This is composed of 24 protein subunits that reach about 20 kilodalton. And they're the body's natural mechanism for transporting and compartmentalizing iron. So it has a hydrated iron oxide core. And there was a lot of work in the 80s actually based around imaging endogenous ferritin, endogenously expressed ferritin. Then people made the logical jump that ferritin could be exploited or modified perhaps to produce enhanced contrast or to act as a contrast agent. So I'm not going to go into this study, but as long ago as 94, this was recognized, and I think this is one of the first studies on this area. But a little bit more recently, it's work from the group of Feraheme, and what people realize is that ferritin, when you reduce the pH, the subunits break apart and separate. And then if you mix in with it something such as a gallium chelate, and then increase the pH, you can entrap these gallium chelates inside of the ferritin structure to create a positive MR contrast agent. And they then attached biotin groups for targeting purposes. And this is a mouse model where they implanted a matrigel plug that became seeded with endothelial cells. They injected a targeting ligand to the endothelial cells that was also conjugated to streptavidin. So when they injected these particles, they accumulated in this matrigel plug, producing this type of contrast that you see here, and is quantified over here when we compare the targeted versus non-targeted agent. So this is approach using natural polymers for cell tracking, where you take Feraheme, which is agent clinically approved as iron replacement therapy, and combine it with heparin and protamine to entrap iron particles inside of this complex. So you can see electron microscopy of these complexes here, and then when they incubate cells with these complexes, you can see these dark spots here indicate iron particles taken up inside the cells. What this graph is measuring is the proliferation of these cells via the MTS assay, and you can see that there's no effect on this measurement of cell function, sorry, not proliferation, cell viability, using this assay, indicating that it doesn't perturb the cells too much. They demonstrated in vivo cell imaging. So a lot of the iron oxides that are clinically approved, such as Ferridex, their synthesis is based on this approach, which uses dextran, so a natural polysaccharide. And if you combine iron 3 chloride, iron 2 chloride with dextran, add ammonium hydroxide, that results in a pH increase, and iron oxide precipitates and is coated by the dextran. So this is an important process. It's the basis of many of the agents that have been, the iron oxide agents that have been clinically approved, which nicely takes me on to the clinical examples of contrast agents formed from polymers. And one of the main examples I'll talk about is iron oxide-based agents. And there have been a lot of different formulations that have been worked on from this review from Crow back in 2006. Virtually all of them have dextran, carboxydextran, some other kind of polymer such as pegoliquid starch as the coating material. And this is just an image of one of these agents being used in humans, where the agent darkens the normal liver tissue, and in this particular case is not taken up by the tumor tissue, so that shows up as being bright on these MR images, allowing diagnosis of cancer. Furthermore, these iron oxides are used for macrophage detection. This is a Cinerem, and this is atherosclerotic rabbit before injection and then 5 days post-injection of these iron oxides. You can see along the aorta of this atherosclerotic rabbit there are little indentations, and so that means accumulation of these iron particles in the atherosclerotic lesions that this rabbit has in its artery. They performed electron microscopy from these lesions and found iron oxides located in the macrophage cells. So this is macrophage imaging. This was subsequently translated to humans. There's still work coming out in this area, but where you see here, this is a patient, the carotid of a patient pre-injection, and then 36 hours post-injection of iron oxide, and you can see various regions of darkening. This was a study where they placed the patients on statins, and over time, on repeated imaging sessions, you see less and less uptake of the iron oxide agents, indicating a therapeutic effect of this of the drug, as read out by the iron oxide agent. So the last thing I'm going to talk about is again these albumin-binding agents. I mentioned them before. This is some clinical data where it's a comparison between this gadofosbacet, it's kind of hard to say, that has this hydrophobic region that binds to albumin, and that then generates longer circulation time for this agent, also increased relaxivity compared to rather more simple gadolinium DCPA chelate. And here are two comparative images from different patients injected with these two agents, and I think you can appreciate increased brightness in the agent, in the patient injected with a long circulating agent, perhaps better visualizations from the leg arteries, and in this case with a shorter circulating agent you can see more contrast here in the kidneys, indicating its excretion. And they also made a number of metrics where radiologists read these images and scored the images, and the long circulating agent that was bound to albumin scored better on a number of them, indicating its superiority over MagnaVist. So just to summarize, there are many different polymers that you can use to create your contrast agents. You can use these polymers as coatings. You can use them as matrices to embed your contrast-generating materials in. They can have nanoparticle properties. They can enhance the contrast that you observe. They're very useful platforms to integrate multiple different types of contrast in one platform, potentially also integrating therapeutics such as the drugs mentioned by the previous speaker, or other types of therapeutics. And there are several agents that are based around polymers that are used in the clinic. So I'd like to thank the people that work for me, my collaborators, my funding, and I'll take any questions at this time.